you want to go back and look at it, or if you're not able to make it, the audio doesn't always come through very well, so they can't hear your guys' questions, which, for better or worse. But they are posted. Dr. Evans, he sent out a link. We'll put a link on the Moodle page, and that link takes you to the playlist of all of the Biol131 SI videos. So, let's dive into chemistry. So you've got elements. Everybody comfortable with what elements are? Right, so you've got elements, and elements make compounds. Your compounds can be diagram. Okay. So if you hold on a second. So we have our matter, right? So basically anything has mass, space, anything. Matter, we can have pure substances, and we can have mixtures. Within our mixture, we have what are called homogeneous or heterogeneous or heterogeneous mixtures. So homogeneous mixtures are going to be the same throughout, whereas heterogeneous will be different. So if you have an homogeneous mixture, triangles and circles, so everything is equally spaced out, and the solution is constant. Now, whereas heterogeneous mixture might look more like this. So you can see distinct differences in the solution. Does that make sense? Okay. Within your pure substances, we have elements and compounds. The elements are what you see on the periodic table. They are the most simplified form of identifiable matter. So that's before we use protons, neutrons, which are ubiquitous across the board. And compounds are combinations of elements. So within our compounds, we can have covalent or molecular compounds, molecule, and ionic compounds. Everybody good there? So your covalent compounds are formed with covalent bonds. That means the sharing of electrons. Whereas your ionic compounds are formed by ionic bonds, and that involves more of the donation, the transfer of electrons. Transfer of electrons. Your covalent compounds are made up of nonmetals, and your ionic compounds are metal with a nonmetal. Does that make sense? Okay. Your ionic compounds are metal with nonmetal, your covalent compounds nonmetals. That being said, what that's really driven by is the electronegativity of the compounds you're working with. So let's talk about why something would bond ionically as opposed to covalent. So if you have, start with chloride, methane, and sodium chloride, we have one atom of sodium, one atom of chloride. How many valence electrons does sodium have? Valence electrons. Any idea? Take one faster. Sodium is right here. How many valence electrons is sodium? One. So if you find sodium, sodium is atomic number 11, which means it has 11 total electrons, but it's in the first group. So it is going to have a valence electron, one valence electron. So we have sodium, and in sodium's outer shell, we have one electron. Does that make sense? We're going to say Na, one electron. How many valence electrons does chlorine have? 7. So chlorine has 7, which means so we say 7. Chlorine, we can say like that. What is, how many electrons do we need to be stable? What is a stable number of valence electrons? 8. So all of your atoms want 8 electrons, except for hydrogen and helium, which want 2. The reason for that is in the outermost shell, the valence shell, of all of the shells, 8 is the maximum number. Some exceptions when you get chemistry, but for the most part, you want 8. In that first shell, there's 2. So let's look at sodium for a minute. Sodium, we said, is atomic number 11. So it's too specific here, but its first shell, it has 2 electrons. Next shell, it has 8 electrons. And in this outermost shell, it has that 1 electron. Don't worry about this. So what happens here is chlorine is very, very strongly attracted to that electron because it's really close to having it. The affinity for that electron is so high that it ends up taking that electron from sodium. So chlorine is stable now with 8. How many valence electrons does sodium now have without that electron? 8. Because we lose this electron, which means its outermost shell with electrons is now the next one in. Now sodium is stable with 8. So that ends up transferring over. And when we lose an electron, what charge do we get? Positive. Lose an electron, electrons carry a negative charge. Lose an electron, you end up with a positive charge. And the chlorine has a negative charge. Ion now, so it's chloride. And so just like magnets, those two will be attracted to one another. Does that make sense? Any questions about that? Okay. Methane, we have carbon. How many valence electrons does carbon have? 4. And how about hydrogen? How many valence electrons does hydrogen have? 1. How many electrons does carbon want to have in its outermost shell? 8. How many electrons does hydrogen want to have? 2. So each hydrogen is going to contribute an electron to each bond. Carbon will contribute an electron to each bond. Just remember, bonds are the sharing of 2 electrons. Hydrogen has half of the electrons it needs. Carbon has half the electrons it needs. So without getting too specific in the electronegativity of it, they both equally want the electrons. They're going to equally share that electron. Question? No. We'll talk about hydrogen bonds in a minute. These are covalent bonds. Hydrogen is bonding, but these aren't hydrogen bonds. So good question. So we end up here is carbon bound to 4 hydrogen. So this is a covalent compound, molecular compound, a molecule with 4 covalent bonds. Any questions on this? Now let's talk about water. So how many valence electrons does oxygen have? Right. So oxygen is in the sixth group. Remember, we're going to forget about everything in the middle when we're counting. So 1, 2, 3, 4, 5, 6, 7, 8. So oxygen has 6. So we have 2 pairs and 2 by themselves. Don't worry about the specifics of that. And hydrogen. So how many valence electrons does hydrogen have? 1. Hydrogen has 1 electron. So hydrogen is going to treat its electron, both of them to make 2 bonds. So we're going to end up with covalent bonds here. Now, oxygen is very electronegative. So as you get into this region of your periodic table, that's increasing electronegativity. So does anyone remember what electronegativity means? What is the definition of electronegativity? Electronegativity is the attraction to an electron. So if something has high electronegativity, it will be more strongly attracted to an electron as opposed to something with low electronegativity. That makes sense. So oxygen has a higher electronegativity than hydrogen. So what's going to happen is rather than equally sharing these electrons, oxygen is going to pull them a little closer to it than it is to the hydrogen. So we would note this like this. Don't worry about representation. What that means is that oxygen is drawing the electrons closer to it. So what do you think happens to the charges if something has the electrons closer to it? Do you think it would be more positive or more negative? More negative. So what we say is this has a partial negative charge. And the hydrogen would have a partial positive charge. Does that make sense? So what's happening here, this unequal distribution of electrons in a covalent bond, that's what we call a polar bond. The unequal sharing of electrons in a covalent bond is a polar bond. Now you can have polar bonds in nonpolar compounds. If we have CO2, CO2 looks like this. Don't worry about the structure. So what's happening here is oxygen is more electronegative than carbon. Carbon and hydrogen are very close electronegativity. So oxygen is going to draw the electrons towards it. But what's happening here is that they're being pulled in equal and opposite directions, which basically cancels the effect out. So although you have polar bonds, because they are equal and opposite, carbon dioxide is a non-polar compound. Does that make sense? Whereas something with water, because of its geometry, you have the oxygen pulling the electrons in the same direction. So we end up with a partial negative and a partial positive region. Yeah. Water is a polar covalent compound. Does that make sense? Right, that's what gives it its polarity. Does that make sense? So when you hear things like the phrase light dissolve light, that refers to polar solutions will be miscible with each other, and non-polar solutions will be, but they won't intermix. Right? That's why water and oil don't mix. Water is a polar solution, whereas oils are a non-polar solution, and so they don't mix. What's happening there is that the partial positive and partial negative regions of those compounds are interacting with one another. You have attractions between the partial positive and the partial negative at different spots, and constantly compound, as opposed to the non-polar solution, which does not have those interactions and therefore won't interact with polar compounds. So it's more energetically favorable for them to be separated. Does that make sense? Yeah? A lot of information. Any questions on that? So that gets us into hydrophobicity and hydrophilicity. So if something is hydrophobic, what does that mean? What's that? Water fearing. The work of the word. Hydrophobia, you're afraid of something. Hydro is water, so it is not like water. It's not going to interact with water. Hydrophilic means what? Water loving. You can break down the word again. Philic, loving. Hydro is water. So, for the most part, I don't think you'll really ever encounter an exception. Polar solutions are hydrophilic. Non-polar will be hydrophilic. Does that make sense? Fantastic. Now let's talk about, stay on the topic of water and talk about hydrogen bonding. So hydrogen bonding is the reaction between hydrogen bound to oxygen, nitrogen, or chlorine to a different atom of oxygen, nitrogen, or chlorine. So we say oxygen, nitrogen, and chlorine because as you get into this region of the periodic table, your electronegativity increases. So what happens here, water, is because oxygen draws those electrons towards it away from the hydrogen, we end up with partial positive and partial negative region, which then allows the oxygen from a different water molecule to interact with its partial negative. Yes. Yes. What do you mean? What are you referring to with the? Yes. Yep. What's that? Yeah. Well, there depends on what you compare it to. That oxygen, and what is unique about oxygen, or what's unique about water, and what gives way to the different properties of water, such as your surface tension, your capillary action, is that water has, so oxygen has two pairs of electrons here, so it can actually interact with two hydrogens. It has two partially negative and two partially positive regions, which lets it create sort of various pseudo structures because it's a 1-to-1 ratio, and that's why water is so effective. Any questions on hydrogen bonding? You talked a little bit about the strength of bonds, so I'm going to cover something you probably haven't seen a lot of, but that you will see soon, whether in bio or chemistry. So we have a ionic bond, and then ion dipole, dipole, and then we have use dipole, I wouldn't worry about writing them down or memorizing them, but it will help you understand what these bonds are. And then we'll think about so hydrogen bonding is a form of dipole-dipole. So a dipole is a partially charged. Ionic bonds, remember, you have a full positive charge and a full negative charge. That's why those are our strongest of the charge interaction. Ion dipole means we have a real charge with a partial charge. And so that's like if we put a sodium ion here, it will be attracted to the partial negative of the oxygen, but not as strongly as it would be to a full negative of the chloride ion. Dipole-dipole, that's what we see with hydrogen bonding. Hydrogen bonding is the strongest form of dipole-dipole interaction, largely because hydrogen, once that electron gets further and further away, that's the only negative part of the hydrogen, so it becomes a strong partial positive charge. Induced dipole, you have a partial positive charge, and what that does is the positive charge is attracted to electrons. So it's going to draw electrons even from things that aren't partially charged. So let's say it's an induced dipole. So basically it creates a partial charge. Does that make sense? Don't worry. Of negative. Right, so if we have this hydrogen that has a partially positive charge, and we put, it really doesn't matter, some compound, it's going to draw the electrons a little bit closer to it because it has a partial positive charge. Right, and what that will do will create what I'll say is a partial negative charge over here, and a partial positive charge over here. No, don't worry about the specific. Basically, I put this up so you can understand the difference between your ionic bonds and your hydrogen bonds and why some are strong and some are weak. Don't worry about the specifics. Moving? Good. Okay. Next, we'll talk a little bit about functional groups. All right. Anybody want to take a stab at what functional groups? If you were to define functional groups, what would you say? Kind of, not really. Yeah, okay. Not really. I like that. So your functional group is a specific region of a compound that determines how it will react. So some examples of functional groups. We have the amino groups. Are other amino groups. Carbonyl group. So carbonyl group, you have things like ketones and aldehydes. That's carboxyl group. This is your carboxylic acid. Hydroxyl groups. Those of you in chemistry know this. OH group. Also alcohol. Phosphate groups. Those of you in chemistry should open that one by now. And last one you might see called hydro group. Knowing these and their exact functions will come into greater detail if you go forward with chemistry and do organic chemistry. We'll focus on a couple of different applied functions of each of these. So, where do you maybe remember seeing any of these in your previous biology curriculum? What you know about the body? Any idea where some of these might be present? Yeah, phosphate groups are in DNA. The other big one for you to know for phosphate groups is in ATP, adenosine triphosphate. Any other ideas about where you've seen these in the body? Very good. So we'll talk, after we finish up this section, we'll talk about your different macromolecules in the body, one of which being amino acids and protein. Amino groups are especially important as they are related to amino acids because the other forms of nitrogen are mostly poisonous. Gaseous nitrogen will kill you, so we get our nitrogen through protein, through the amino groups on the protein. Yeah. Hydroxy groups are everywhere, always. So I'll show you a couple different reactions. Hydroxy groups are everywhere. If you drink them, you do fun stuff. Anyone have any ideas about ketones? Or ketones, ketones, maybe different diets that have to do with ketones? Keto diet. So when you break down fats, you produce ketones. And that's what the keto diet is all about, breaking down fats as an energy source instead of sugar. When you break down fats, you produce ketones. So that's how they know if you're undergoing ketolysis. They can test to see if you're breaking down fats. Any questions on this stuff? I would be able to recognize this might be a good one for her to put a map in, because they can be tricky to memorize and draw, putting compounds, but it's a little bit easier recognizing the name and where it goes, especially if you can rule other ones out, right? So if you have sulfhydryl group and you know there's sulfur in it, hydroxy group, you know there's hydrogen and oxygen. A phosphate group has phosphorus. For the most part, some of them are tricky, but they're somewhat simple to be through. All right. This is so what are lipids? That's one example of a lipid. The lipids are your fats, oils, steroids, and phospholipids. Do you think lipids are hydrophobic or hydrophilic? Hydrophobic. Steroids are important in cellular signaling. Hormones are based off of steroids. What is important about phospholipids? Membrane. Are phospholipids hydrophobic or hydrophilic? So that's why we have the two layers of our membrane, so that we can have hydrophilic outside and a hydrophilic inside, but still have the self-contained phospholipid bilayer, which we'll talk about more in the next section. We'll talk about more stuff. Fats are glycerol plus 3 fatty acids. Glycerol plus 3 fatty acids. So, glycerol looks like this. And your fatty acids look like this. Fatty acids, they're an acid. They have this group, which, if you remember from what I just erased, is a carboxylic acid group. So what happens here is that the glycerol binds with the fatty acids through a dehydration or a condensation reaction. So what that means is that we pull water out to bring these together. So this oxygen will bind with that carbon. It does not matter which hydrogen you imagine, which oxygen you imagine, it still produces the same stuff. So that would be a dehydration reaction. If we break it apart, if we remove this fatty acid from the glycerol, that would need to absorb water, which is the opposite direction. And if we are breaking down water, we go to hydrolysis reaction. Hydro meaning water, lysis meaning breaking down. That's why the The next section will talk about lysosomes, how your organelles that break down things in the cell. So the integration reaction is reverse hydrolysis. So within our fats, you have saturated fats and unsaturated fats. We have glycerol. This is a branch fragment. And we have our three fatty acids. We can also have it look more like this. With double bond. One of these is a saturated fat, the other is an unsaturated fat. Anybody want to take a guess at which is which? Yes. So this is our saturated fat. So saturated meaning saturated with hydrogen. So it has as many hydrogen as possible. Unsaturated is not saturated with hydrogen because when we add a double bond, we lose the ability to bind to hydrogen. Chemistry, we have to do that. So what happens is that because these are all uniform, they're able to pack closely together. They pack closely together, that's why our saturated fats are solid at room temperature, and our unsaturated fats are liquid. So butter is an example of saturated fat, whereas the oils are unsaturated fats. What we have found is that organisms that live in the cold weather, their cell membranes actually have more unsaturated lipids, phospholipids. They give the membrane more fluidity at cold temperatures. Any questions on lipids and fats? All right, carbohydrates. Lipids are going to be carbon, hydrogen, oxygen, carbon. So proteins will be carbon, hydrogen, oxygen, nitrogen, nucleic acids will be carbon, hydrogen, oxygen, nitrogen. And these are your five most important organic elements. So who knows what carbohydrates are? Energy source, those are our sugars. So we can have simple sugars. We can have monosaccharides, disaccharides, and polysaccharides. So monosaccharides are one simple sugar unit. And these will either be non-hexose or pentose sugars. So we can think of that as a monosaccharide. So anybody know the most common, most important monosaccharide? Glucose. So glucose is sort of your pivotal example of a monosaccharide. Disaccharides you can think of as two of these together. So say the example glucose. An example of your disaccharide is sucrose. Sucrose is actually glucose and fructose together. polysaccharides are large chains of these sugar compounds. On and on. The polysaccharides are useful in energy storage. They're also useful for some structural components. So, some examples of polysaccharides that have to do with structure. So, we'll start with energy. So, energy storage, we have things like starch and glycogen. Glycogen is most common in humans. We use that to store glucose. The large branched chains of glucose are glycogen, and that's what we store in our muscles. That's why if you are an athlete and carb up the day before, it is you put sugars in your body, you store it as glycogen, and then as you are exercising, your body breaks that down. And structural, we have things like cellulose and anybody know the difference or know where you might find one or the other? Mostly in plants. Plants have their cell wall. Chitin is mostly in things like insects with exoskeletons. We also find it in bacterial cell walls as well. Cellulose is fiber. We don't break it down. Unlike our energy storage polysaccharides, we don't break that down actually at all. Any questions on carbohydrates? Yes. So starch is one of the energy storage compounds, where cellulose is more structural. All right, good. With carbohydrates. Fantastic. Proteins. They are our source of nitrogen for the body and building blocks of a lot of our biological structures. Proteins are composed of what? Made up of amino acids. Amino acids are building blocks of protein. So similar to something like glucose to glycogen, amino acids are. We have 20 very common amino acids in the body that builds up all of our various proteins. With proteins, you should know structure. We have primary structures, secondary, tertiary, and quaternary. Anybody want to take a stab at the definition of these various structures? So our primary structures are the chain of amino acids. Chains of amino acids. Secondary structures are the substructures driven by hydrogen bonding. Tertiary structures are driven by interactions between R groups protein, each amino acid has a unique R group that it can be interacting. Tertiary structures are the most advanced structure of a single chain of amino acids. If here we have just a chain, our secondary structure, exactly what these structures are, doesn't matter. As you get more into your courses, you'll learn what each of those are. Tertiary structures are the final product of a single chain. And quaternary structures are the interaction between polypeptide subunits. Polypeptide subunits, meaning these chains. Quaternary structures are primarily driven by hydrophobic and hydrophilic interactions. So if we have a tertiary structure that has a hydrophilic region and a hydrophobic region, if it's in an aqueous solution, it may find another subunit that is similar, so that the hydrophobic region is, I'll say, protected from the solution outside. An example of a quaternary structure would be hemoglobin. Hemoglobin has 4 different subunits that come together and basically cover the hydrophobic region. Does that make sense? Any questions on protein? No. Last thing to talk about proteins are your enzymes. So enzymes are protein catalysts, so they help promote reactions. What you should know about enzymes is the induced fit versus lock-in key model. Induced fit versus lock-in key model. A lock-in key is primarily an older model where the idea was that, similar to a lock and a key, if you have one enzyme to one substrate, one enzyme, and the enzyme would match it perfectly. One enzyme to one substrate. Induced fit model is sort of the leading model with enzymes and substrates, and basically says that enzymes and substrates will change slightly when coming together. As the name suggests, it's an induced fit. It creates that fit. Does that make sense? So enzymes promote biological functions and allows biological reactions to happen. Real quick, with your enzymes and basically any biological reactions, you can have different forms of inhibition. You have competitive or non-competitive inhibition. So what that means is that if an inhibitor is binding in the active site, preventing that reaction from happening, that would be a competitive inhibitor. Whereas if it belongs somewhere else and causes a structural change, preventing the reaction, that would be non-competitive. So it's competitive if it's in the same space, non-competitive if it's in a different region. Does that make sense? All right, real quick, before we finish up, we'll touch slightly on nucleic acids. So what are your nucleic acids? DNA and RNA. What are nucleic acids made up of? Nucleotides. Proteins are made up of amino acids. Ribosomes are built from proteins. or from RNA. So nucleotides. So similar to how a glucose builds up glycogen, different nucleotides build up DNA, similar with proteins. With proteins, we have, for the most part, 20 different amino acids. For nucleic acids, we have 4 nucleotides. And you'll see it abbreviated as A's, C's, C's, and G's. So A binds to G, B binds to G. This is the same A that you see in ATP. You have adenosine triphosphate. It's also present in your nucleic acids. And we actually have, you can also have like GTP, or with cellular pigments that would be just as effective for energy storage. We just don't use it. DNA built basically is the blueprint to what we are. Over the course of evolution, DNA changes. Changes driven by mutations. And when mutations are favorable in the environment, they become more prevalent, and that species eventually evolves. So mutations are changes in this ACCD sequence that cause a change in the physiology of that organism. Any questions on nucleic acid? Any questions on the multitude of things that we talked about in the last hour? No? All right. You guys have a good night and good luck on your exam. Frankly, I didn't think we were going to get through it all.